everybody, and welcome to the second day of our workshop on fish spawning aggregations and fishers' knowledge. We've had a full day yesterday with 2 sessions, and then this day is session 3 and session 4. Very happy to see you here, and I hope you're going to enjoy the workshop. I'm going to hand over quickly to Fadila, who's going to give us a little bit of logistical comments on running the workshop, and then it'll come back to me for the session 3 opening. Thank you.
Good morning, everyone. Good evening, good night, wherever you are. Welcome to day 2 of our training workshop. Just a little bit of logistics and housekeeping. We encourage you to use the Q&A button if you have any questions. We encourage you to post any questions throughout the session, and that could be found on the bottom ribbon of your Zoom panel. You can also use the chat to generally chat with other attendees or put other comments. However, if you have questions, please put it in the Q&A box, and we will try to address this at the end when we have a 30-minute session. And if we have run out of time, we will answer any other questions via text or follow up via email. Back to you, Melita.
Thank you, Fadila. So good morning, good afternoon, hello, wherever you are in the world. So this session, session 3, is called Applied Research, Designing Your Approach, which I'm going to be moderating. My name is Melita Samoilys. I'm from Cordio, East Africa, based in Kenya, and I'm also the co-chair of the Species Specialist Group, the Snappers and Associated Families, co-chair with Ken Lindemann. So in this session, we cover some of the practical how-to questions for FSA research. That's FSAs, fish spawning aggregations. And we provide guidance on designing your research and conservation programs. Fishers can play a key role in FSA site identification and cooperative research, particularly where there's little known in your area. So we'll hear some of the common verification and monitoring techniques, such as underwater visual surveys, documenting reproductive status, tagging and acoustic telemetry, and monitoring the dynamics of fish movements to and within aggregation sites. Examples of how FSA research results have helped design conservation action will also be discussed. We've got some great guest presenters who'll give us very short presentations, and they've got research experience from widely different regions, such as Dr. Rick Nemeth in the US Virgin Islands in the Caribbean, Dr. Alec Hughes in Solomon Islands in the Pacific. And as well, we've got detailed talks on the methods for FSA research from Dr. Kevin Rhodes, who's worked on FSAs extensively in the Federated States of Micronesia in the Pacific Ocean. And we have Dr. Will Heyman, who's one of the other leads of this workshop, and he's worked extensively in the Western Atlantic and Caribbean. So I'm going to hand over now to Will, and just give you a very brief intro on Will, particularly for those of you who weren't here yesterday, where you would have heard him a lot. He has a very strong academic record from positions at universities and international NGOs, and he's done incredible work on fish spawning aggregations over many years. So he's got a huge accumulation of experience and knowledge. He works with a high focus on cooperative research with fishers and also scientists and managers, and this has largely been in the Western Atlantic. Through this, he's fostered collaborative marine spatial planning, including the creation of 20 marine reserves that support both marine conservation and livelihoods. So over to you, Will.
Designing your research, monitoring, and conservation approach. This talk offers a general framework for the stages from discovery to sustainable management of fish spawning aggregations. Various stages in management, starting with discovery, Fishermen will often discover this place, and as we've talked about, start fishing. Makes sense to me. But if they are concerned or interested, they may indeed alert conservation managers or scientists to that discovery and ideally work together to begin to characterize that site. Ultimately, to prove it's an aggregation site will require verification, at which point it may lead to management planning and ultimately to monitoring. And you can see there's some overlap in all of these activities. It's a nonlinear process. But if things go well, management could be enacted, either a marine protected area or seasonal closure or other measures. And ideally, enforcement might ensue, which could lead to sustainable management. This is an example from Don de Maria in Florida. Don here took this picture back in the late 80s, spearfishing with his friends for Goliath grouper on a spawning aggregation. Years later, Don became a very important conservation leader. He participated in cooperative research, both in Florida and Palau with Pat Collin. He was instrumental in getting Goliath grouper closed to fishing. He's been working on FSA management, including the declaration of the Tortugas Reserve, Riley's Hump in the Florida Keys. He was one of the founders of the Big Fish Initiative. He won the first GMA award in 2004 and continues as a conservation leader in Florida and around the world. When aggregations are identified, there's typically indirect evidence that it may be occurring. These could include, as we said, a large, dense group of fish, potentially fishermen fishing on them. But underwater, you might observe courtship behaviors and colorations like you do in the picture on the right. You could also hear or record courtship-associated sounds. Or underwater or at a landing site, see gravid females with swollen abdomens. Underwater, you might see male-male aggression, chasing, fighting, or elevated gonosomatic indices, again, from landings data. But a note of caution, not every large group of fish is a spawning aggregation, so it's important to verify that. And there's very few techniques to actually verify a spawning aggregation. One is the undisputed observation or video recording of actual spawning, gamete release, but also females with hydrated gonads, hydrated oocytes, or the presence of post-ovulatory follicles in the ovaries of aggregating females, a technique that requires histology. And you'll hear more about all of these techniques in the subsequent talks in this section from Kevin Rhodes, Melita Samoilys, myself, and Rick Nemeth. There's very many manuals and resources out there available for your use, and you can find many of these on the SURFA website, probably the most comprehensive existing resource. But the new Big Fish Initiative website also has a tremendous amount of information, not only manuals and resources, but also videos and all kinds of things. And we'll shortly be housing the results from this workshop, as well as other resources for your use. When you're designing this approach, make sure to choose methods that are appropriate, things that are management focused, things that you can afford, and that are designed for the purposes that you're trying to accomplish. You might need different techniques for characterization than you need for monitoring. You might need different techniques depending on your research questions, if that's what you're after. And there's various common techniques that have been used. This isn't comprehensive, but some of these methods are listed out, including the type of information that could be generated. For example, here are methods showing which could document indirect evidence of an aggregation versus direct evidence. And here are methods characterized by their utility for discovery, characterization, verification, monitoring, or research. So you're going to hear a bit more about all of these techniques in the subsequent talks, information about fisher interviews and examples, diver surveys, underwater visual assessments, a little bit about site mapping, as well as the use of fish tagging, acoustic telemetry, and active acoustics. When you get into long-term monitoring, there are two ways to go about it, one with repeated site visits, the other with long-term in situ instruments for remote sensing. In terms of repeated site visits, the most common is underwater visual surveys. Size, age, frequency, sex ratios, and reproductive status can all be gathered at landing sites or dockside. But it's important to remember, in my opinion, the goal of this is to actually support management. So you're looking for trends in abundance, in time and space, and according to environmental drivers. You could also, and importantly, document site use in terms of fishing and or potentially tourism or other uses, again, to support management. In addition to site visits, you might use a variety of remote sensing techniques, putting sensors underwater at the aggregation site, including passive hydroacoustic recorders that could document courtship sounds or boat traffic. You might use acoustic Doppler current meters or other types of current meters to document the speed and direction of currents over time, or a multi-parameter sonde to look at time series trends in conductivity, temperature, oxygen, or other physical parameters. You might use acoustic telemetry receivers that could record the presence of tagged fish as they go by, fixed underwater video cameras, and again, there are details being provided and examples in subsequent talks. In terms of management options, I want to remind us all that working cooperatively with fishers throughout the process of planning and management is absolutely key. You might wind up creating marine protected areas for the aggregation sites or the migratory routes, could implement seasonal closures, gear restrictions, harvest quotas or allocations, total species bans, rotating closures, rights-based management initiatives, or, as Yvonne Sadovi indicated in 2016, it may be best to just leave these places undiscovered and uncharacterized. Interestingly, there's been some new developments on ecotourism. You saw the talk by Richard Barnan and the example in Palau, but really, a combination of these management measures is typically the best bet. In my opinion, it's always best to keep it simple. This is a lovely example from the Coral Triangle, where repeated UVC measurements, along with community members along permanent transects, were able to detect temporal trends in the FSA populations. It built community support for FSA closures in this area. Really nice, simple example, inexpensive, very, very powerful. As I mentioned in Palau, the use of ecotourism as a tool has various benefits. It might slow fishing if that's occurring, just because of the presence of divers. It might generate funds for management and certainly can raise awareness. Obviously, there are concerns. You have to be aware of carrying capacity. You need codes of conduct and research on the impacts of dive tourism on FSAs. But I like to say, and there's more research to be done on this, it's better to be disturbed than to be dead. There are a variety of best practices to build capacity for monitoring and management. The most important of which, or probably the most powerful, are training workshops. This is one we did back in Belize. Here's Kevin Rhodes, who helped me put this together. The two of us were leads on this. This was a training that includes 60 people from 9 countries and really spawned a tremendous amount of conservation effort. In fact, most of the people that you see in this photograph are still involved with the conservation and monitoring of FSAs all over the region. Fisher exchanges can also be very, very valuable, taking fishers from one area to another. We did one between Belize and Indonesia, which was very instrumental in helping raise awareness. But awareness is key, and using media and outreach is absolutely critical to get more people aware of this important phenomenon and the need to manage FSAs. And I gotta say that the Big Fish Initiative represents a wonderful example about how this can be done. Thanks very much for your time.
Wonderful, Will. Thank you so much. That was absolutely spot on. So the next talk, moving right along, is actually by me, and it's on conducting fisher interviews, so around fisher knowledge and catch landings to find and verify FSAs. Over to you, Fadila. Thanks. Welcome, everybody. In this talk, I'm going to be talking about fisher interviews and catch landings data to verify FSAs. So what I'd like to talk about is some of the first steps that we often take in a research or conservation project to find and then verify FSAs. When you're starting from scratch, it's hard to know where to begin. So I'm going to give an introduction to 3 sources of information or data that are very useful in helping you to start your research on fish spawning aggregations, and I'll abbreviate that to FSAs. Firstly, choosing the species. Find out what's published on the species that you might be interested in working on. That's a real first step. The second thing is I'm going to talk about fishers' knowledge, or running interviews with fishers to understand what local knowledge they might have on FSAs. You've heard about FK already, sometimes called LEK, local ecological knowledge, and you'll hear much more about that in session 4 later today. The other data that's extremely helpful, useful and important is fishery catch landings data, so I'll talk about that. And then I'll finish briefly with discussing some potential local management or conservation measures to be taken up with fishers in coastal communities. So which species? Some useful criteria to guide your initial steps. What's your interest in certain taxa? Which species are you particularly interested in as a researcher? Are there vulnerable or endangered species you'd like to know about? And then what anecdotal local reports on fish aggregations have you heard? And have you come across any high landings of a species at a particular time in the local fishery? We do, of course, strongly encourage you to choose the families in our SSG because there are large parts of the globe where we don't know anything about their spawning aggregations, such as in Eastern Africa. So as I said, to choose your species, read the literature. There's a huge amount of literature out there, and because nothing's been done in your area, it doesn't mean that that species hasn't been studied somewhere else. When we did this back over 10 years ago, working with Jan Robinson and our other colleagues, we looked at what was published in the Indo-Pacific to guide us on what might be forming spawning aggregations in Eastern Africa, particularly in Seychelles, Kenya and Zanzibar. And so this is an example I provide of what we found on snappers and emperors at that time. And this is covered in this book that's illustrated there, that Jan Robinson and I produced in 2013. There's several chapters and different authors. And I will make that available on Hoover, on the site. So when you find that evidence in those papers, you need to make sure that the verification criteria follow the published criteria that have been agreed. Direct signs of spawning, such as observations of gamete release or hydrated ovaries, and then indirect signs, things like patterns in the gonadosomatic index, GSI, or male colour changes and territorial behaviour when watching spawning underwater, observations of gravid females, and finally then a threefold increase in abundance measured at a site. And you'll hear more about that in Kevin Rhodes' talks just a little bit later in this session. So turning to fishers' knowledge, which will definitely be covered in more detail in session 4, I just want to touch on things and really give you some examples from Eastern Africa. It is a key approach to gathering information on FSAs. If they have knowledge of the occurrence of spawning aggregations, it can provide a vital source of information. So it's a great place to start where you've got no information at all. So talking to fishers and using questionnaires has several ethical rules of social science, of course. This is in terms of intellectual respect, in terms of rights, participation, consent, etc. And you can learn more about that from the IUCN guidelines that Winston Cowie is going to be talking about in session 4. One thing to be aware of in such questionnaires, and these are designed for one fisher at a time, is that you do want to bias your sampling in terms of choosing your fishers. So I've had difficulty sometimes conveying that with my teams in Kenya, because we actually want key informants. We want, say, the elder fishers who've got more experience, more years, and therefore more knowledge. So that's fine. It doesn't mean the questionnaire is being biased. So obviously, having decided you're going to do that, you need to think about the design of the questionnaire. And this is also covered in session 4, so I won't go into too much detail on the design. Here I'm illustrating the Fisher's Knowledge Questionnaire that we used in Kenya and then modified further in Zanzibar. It's available in that Robinson and Samoilys 2013 book on FSA research and management in the Western Indian Ocean. It was broad to cover the fishers' information and perceptions on catches, trends in catch, gears, effort and fishing grounds. So here's just a couple of the pages in the questionnaire. So these are fairly standard fishery survey variables. When starting the questions, it's really important to do a thorough introduction as to why you're doing this work, and that takes time. You can't rush it. So you have to build trust, and you need materials to show what you mean by spawning aggregations. And a good way to do this is actually involve the fishers in the research, those that have knowledge. We've found in Eastern Africa that participatory research that leans on local knowledge can be very effective in doing the work and spreading awareness and implementing conservation action. This page covers knowledge on the FSA itself, and it requires quite a lot of support material, especially photographs and maps, to illustrate what we mean by spawning aggregations. Video would be great too, which we didn't have it at that time. And this gives you some examples of the questions you can use to drill down into the fisher's knowledge of SSAs, like what did they see in terms of which species, which months, which years, did they notice the moon phase, and particularly have they fished them. We found this very instructive with basket trap fishers who were targeting rabbitfish aggregations. Now I'm moving on to catch landings data and surveys, and these provide a really important addition to fishers' knowledge questionnaire surveys. They can help you confirm the species and the timing and the location of FSAs, and it's most easily done looking at catches at landing sites. You can have first-hand observations of large numbers than usual of a species being landed, combined with highly gravid females, for example. This can provide good evidence of the timing of spawning, but of course it doesn't provide evidence they were in a spawning aggregation. So it's exploratory, and the data I'm showing here is data we collected on rabbitfish in southern Kenya, on the left there, looking for aggregations, evidence of aggregations, and the histograms are the fisher interviews. So when they reported the aggregations were occurring, which are largely in November, December, versus the CPUE or catch landings, which reports a peak in August. So you can see they don't match. And the small graph on the right was the number of landed Epinephelus lanceolatus giant grouper versus lunar day, giving us an indication of just after full moon spawning. A further slide on catch landings, just to stress really the importance of being able to do simple visual observations of gonads in catches. This photograph illustrates part of our early evidence of a grouper spawning aggregation, Epinephrus fasciogutatus, which combined with fisher interviews led us to 2 aggregations which occurred on the full moon. Further confirmation of spawning aggregations can then be done. We used this on scuba, and you're going to hear more about that from Kevin Rhodes shortly. So to finish, I'd just like to flag locally managed marine areas, or LMMAs, as a conservation and management approach. These are led by fishing communities, and they've proliferated in the Indo-Pacific. They started in Fiji and the Philippines in the 80s and 90s, and they've spread to the Western Indian Ocean in the early 2000s, where they've really taken off, especially in Madagascar and Kenya. And the key things here is that they have high potential to contribute to OECMs, other effective conservation measures, and therefore the 30 by 30 target. But conserving FSAs with fishers' knowledge using LMMAs is a very powerful approach. Thanks for listening, and see you soon. Okay, thanks for listening. The next talk is from Will Heyman again, and he's going to be talking about site characterization and mapping. Over to you, Will.
Site characterization and mapping. Mapping is an iterative process with more information during the process between characterization all the way through monitoring. Your maps get better and better with more information. You can start with sketch maps, but ultimately you're probably going to need bathymetry maps. And ultimately for monitoring, it'll be necessary to map fish density or fish counts in space and time. Lots of ways to go about mapping. Aggregation sites, fishers' knowledge, satellite images, aerial photographs, bathymetric charts, Google Earth, and data available in fish finders and or aftermarket online. Lots of tools to make maps, sketch maps, making bathymetric maps, various techniques available. I'm going to tell you about one called adaptive bathymetric mapping, and offer a few ideas about how to do fish count density maps using underwater visual census. If you're going to make maps, start simple. Here's an aggregation site shown from both a satellite image and an aerial photograph. Keep that image in mind. I'm going to come back to it in a minute. Here's a sketch map that Pat Collin made back in the early 90s of a Nassau aggregation site, showing the relationship between the aggregation site, some benthic habitat, and the shelf edge in 2 consecutive years, different places. This map of another site shows the dynamic whereby the aggregation was up at the break in the afternoon, but at the time of spawning, the aggregation had moved into deeper water further offshore. Very simple, very valuable, very powerful. This technique is called adaptive bathymetric mapping. It essentially uses a single beam sonar with an integrated GPS, otherwise known as a logging fish finder you can buy off the shelf. Data are collected in a series of transects, downloaded, cleaned, and interpolated using some kind of GIS system. Why do we call it adaptive? Is because data collection concentrates on those places where it's most needed for efficiency. So for example, very little data is needed to map this flat area. And we know where that flat area is from 2 images ago. The geo-referenced satellite image told us where the shelf edge was. So very little data are needed here. A lot more data are needed near the shelf break. So that's the initial design. If more data is needed, you just go back and get more and reinterpolate to make your map better and better over time. This has been used to map dozens of aggregation sites all over the world. For monitoring, though, you really need to map fish density or fish counts in space and time in reference to the bathymetry. And the most common method to do so has been underwater visual census. And ideally, those should be done on the exact same transect each time so that counts are accurate and repeatable. Here's a sketch map showing where transects are designed, and you're going to hear more about this in a minute. So let me just move on. Another way to do that, another way to map density is something called the GPS density survey that Pat Collin developed, whereby divers swim along a transect with a towed floating GPS. Count data and the time data can be integrated to show counts by space to calculate density and ultimately extrapolated to total count. Here's an example of the output showing density of two different species in the same place on the same day. Counts plotted in space and time in relation to environmental conditions offer you good data for monitoring. Couple cautionary notes: very much variability in all of the dynamics, and SSG species are typically much more variable in space and time than ceranids, for which many of these methods have been developed. Also complicating issues is trying to map out and monitor multi-species aggregations. And final note of caution, site maps should probably be kept confidential until management is well underway. Thanks so much.
Great talk from Dr. Will Heyman there. So now we're going to move on to one of our guest speakers, Dr. Kevin Rhodes. Kevin has worked extensively in Ponape, Federated States of Micronesia, on fish spawning aggregations of various species.
Methods to improve area and temporal management at a multi-species spawning site. Ponape State Micronesia is a group of small islands and atolls located in the Western Pacific. The main island Ponape is home to around 30,000 inhabitants, many of whom are relying on fishing for subsistence and income. The island is heavily overfished and utilizes nighttime spearfishing as its main fishing activity. Thus, there is a need to gather data on aggregation locations, timing, and species to help improve the fate of both fish and fishers. Local ecological knowledge has provided information on a number of potential aggregation sites, with 23 individual areas identified for grouper, snapper, or emperors. We may assume that some aggregations have been lost to fishing prior to protective measures being put in place. In 1986, Ponape State created the first aggregation-based legislation to protect groupers during perceived spawning times. In Ponape, local ecological knowledge states that aggregations of grouper and other species form when the breadfruit ripens. As such, legislators enacted a March through April sales ban on all groupers based on this perceived linkage. Subsequently, in 1995, Ponape State created the small-scale Kepra Marine Sanctuary around a multi-species grouper spawning aggregation site located near Kepra Island on the Outer Barrier Reef. At the time, no formal records of the aggregation fishery or site monitoring had been done. Thus, the sanctuary design was based solely on local ecological knowledge. However, unbeknownst to the legislators, the initial sanctuary provided area protection for only one of the three aggregating grouper species. Moreover, the northern boundary provided a useful marker for fishers to anchor to target the most vulnerable of the three species, the camouflage grouper Epinephelus polyficatium. Beginning in 1998 and 1999, and using information provided by local Marine Resource Agency staff, the first formal investigation of the site began using underwater visual census, or UVC. Initial assessment showed that each of the three species is located along the outer reef wall that continues down the slope to beyond 200 meters. Only the square-tailed coral grouper, Plectropomus areolatus, utilized the reef flat, beginning at approximately 15 meters depth. Transects were thus placed along the wall in a manner to traverse each aggregation and at the wall flat interface to capture coral grouper in the shallow sections. Monitors recorded abundance and fish length within each transect out to 15 meters, with 5 dives needed in total over 2 days to complete the monitoring task. In this slide, monitored areas are represented by grids. For the Kepra site, UVC was conducted monthly in both years from January to May, starting 2 weeks before and up to the day after full moon. Those initial assessments help delineate the aggregations of each species, identify trends in the arrival and departure times, and estimate general densities within each of the three aggregations. UVC also provided general estimates of the total aggregation area. From that knowledge, we were able to develop a structured monitoring protocol in 2001 using fixed belt transects that are still in use 23 years later. In 2014, we divided transects into 10-meter sections and made counts of each species within each section from 10 days prior to and up to full moon. This effort provided sufficient detail to identify fine-scale changes in density, location, arrival, and departure times. These combined efforts provided legislators with sufficient information to encapsulate all three aggregations and extend the sanctuary an additional 1.6 kilometers to the north. Doing so provided greater protection against fishing for all three species aggregations and allowed the inclusion of a 1,000-meter buffer to the north. By monitoring densities over the years, we were able to determine the seasonality of aggregation formation, including peak times for monitoring. Identifying aggregation formation against winter solstice allowed for more accurate predictions of peak aggregation formation. Using this data, we were able to convince Pohnpei State to extend its seasonal sales ban from March and April to January through May. In summary, systematic and standardized underwater visual senses allowed researchers the ability to provide decision makers with the information they need to enact effective and informed temporal and area-based management. These efforts have paid off as the most recent surveys have shown an increase in densities and abundance for all three species as well as other groupers targeted by the fishery.
Thank you so much, Kevin. And we're going to hear another talk from Kevin, and this time it's looking at fisheries-dependent survey methods for FSAs. Back to Kevin.
For this presentation, I would like to introduce some basic techniques used to identify spawning sites and times for developing management protocols and better understanding fish life history. Identifying and characterizing fish spawning aggregations, or FSA, often starts with fishery-dependent methods. As the name implies, these methods rely on observations and/or sampling of catch at the spawning site, at markets, or at landing sites where captured fish are available for sale or trade. When starting an FSA monitoring protocol, these methods can provide valuable and complementary information on the aggregation that serve as a baseline for future comparisons. Where spawning sites and times are unknown, working with market owners and fishers can lead to site identification where complementary fisheries-independent methods can be applied. Fishers are often available and willing to provide information on fishing times, gear use, and expenditures. Fishing locations are often more difficult to collect, but over time this information may be shared with researchers once trust is established. Let's start with our main premise. Fishing on spawning aggregations typically has severe negative long-term impacts to fishers, fishing communities, markets, and of course the fish populations being targeted. The use of underwater visual assessments can provide direct evidence of these impacts as reductions in fish abundance and density and a reduction in fish size. However, in many instances, direct observations are not possible due to the depth or the remoteness of the aggregation, as two examples. The next three slides show changes in catch, income, and effort over a 10-year time period using fishery-dependent methods. In this example, fishery-dependent methods identified reductions in catch per unit effort, or the number or weight of fish that can be taken in a given time period. By collecting fishery-dependent economic data, losses in fisher income can also be identified. Similarly, an increase in fishing time for the same catch is often a reflection of an impacted fishery. For fish, FSA fishing consistently results in a loss of fish in annual reproductive output, or the total amount of eggs spawned over a given year. At the most basic level, fish length and weight are two measures that are easy to obtain and that can later be combined with other details to gain further insight into the species and its life history. Wherever possible, fish weight and length should be the first measures collected to serve as a baseline and gauge changes over time. Non-invasive methods are often needed when fish are sold whole, and there is a reluctance by fishers or market owners to allow researchers to alter the appearance of the fish by extracting certain tissues. Some tissues, such as whole gonads, are usually required to reliably determine stages of reproductive development or fecundity, which is the number of eggs a particular female is carrying. Otoliths, which serve as a record of fish age, are used to establish the age at maturity and rate of growth. Gathering either of these tissues requires opening the fish and altering its appearance. Where fish are being filleted, these tissues may become freely available, making landing sites a good option for data collection. Alternative non-invasive options for aging include fish scales and fish spines. Section gonads help researchers to identify stages of maturity. Spawning times and reproductive seasonality can be determined by the ratio of gonad weight to body weight, referred to as the GSI. During spawning times, gonad weight increases, elevating the GSI and allowing identification of spawning seasons. Seasonal restrictions on catch, sale, or possession can be developed based on these results. Conveying this information to fishers can help researchers and managers gain trust and may create partnerships within the fishing community. Working closely with fishers and including them in your research is one of the best ways to pass knowledge to them and in return gain knowledge from their experience. Over time, these relationships can provide information on other spawning sites and species and assist in developing informed and appropriate management protocols. There's a lot of information available on the web and in publications that are readily available. Here we provide links to more information on otolith extraction and gonad extraction and processing.
Great. Thanks so much for that, Kevin. So the next talk we have is from Dr. Rick Nemeth. He's a professor at the University of the Virgin Islands. He's worked on fish spawning aggregations for many years, and these have spanned sites from Bermuda to Bonaire, to Micronesia and French Polynesia. So his work, he's particularly known for helping us understand the spatial and temporal patterns of movements around FSAs to improve conservation and management. And he's engaged with fishers throughout his career to achieve successful conservation goals. So over to Rick.
Hello, my name is Rick Nemeth, and I'm a research professor at the Center for Marine and Environmental Studies at the University of the Virgin Islands in St. Thomas. I've been working on fish spawning aggregations for the past 25 years in an effort to better understand the patterns of fish movement around spawning sites for conservation and sustainable fisheries. Before I start my presentation, I want to thank all the people who have been involved in these projects over the decades. Funding was provided by NOAA and the National Science Foundation, and I'd like to give special thanks to all the fishers on St. Croix, St. Thomas, and St. John who supported this research with their knowledge of fishing and fish behavior. Finally, special thanks to divers and field crews who participated over the years to make this work successful. Today, I'll focus on research that has been done on 3 species of snapper in the U.S. Virgin Islands: mutton, cubera, and dog snapper. This 3D bathymetric map shows U.S. and British Virgin Islands. The colored symbols represent known spawning aggregation sites where we have seen hundreds to thousands of snapper aggregating. The yellow triangles represent mutton snapper spawning sites located near Tampo, Germanic Bank, and on St. Croix. The stars and circles represent dog and Kubera snapper spawning aggregation sites. So information from several acoustic tagging studies found that mutton snapper spawning season goes from March to August, with peaks from April to July. Kubera peaks spawning in June to September, and dog snapper actually spawn year-round, but with higher activity from January to April. Before I talk about the spatial patterns, I would like to provide an overview of the spatial and temporal structure of spawning aggregation sites. Evidence from several species that aggregate to spawn shows similarities in their space use. The spawning site is a fairly small area of less than 1 kilometer squared where gamete release can be observed. The courtship arena around the spawning site and is less than 10 kilometers squared typically. And it's where courtship colorations and behaviors can be seen, as well as territorial aggression. And fish occupy this area in the hours before spawning during the week of the lunar cycle. The staging area shown in red varies from 10 to 100 kilometers squared and is occupied daily by some species during the spawning season, and is used for feeding, resting, occupying cleaning stations, and also important for guiding first-time naive spawners to the spawning site. I've highlighted the staging area in red because I believe it's the most important area for defining boundaries of fishery protected areas. The functional migration area includes all the habitats that individuals use as they migrate from their home sites to the spawning site. And finally, the largest area, the catchment area, is used by all fish from a spawning site. It's also a fish defined as a fish stock, which is the basic unit for fisheries management. So here are 2 examples of staging areas shown as these colored polygons for Kubera snapper on the upper panel and dog snapper on the lower panel. Around the Germanic Bank seasonal closed area, which is shown as the orange rectangle. The small dots represent acoustic receivers used to track fish with transmitters. The darker blue areas represent where tagged fish spent most of their time. You can see that Kubera and dog snapper use slightly different areas during the spawning season, but areas are fairly similar in size of about 2 kilometers squared. The migration pathways for these 2 species is quite different. Kubera snapper migrate to and from the spawning site mostly from the east, while dog snapper migrate mostly from the west. Migration pathways are also important for management considerations since they concentrate fish to specific areas. Now let's look at mutton snapper in the U.S. Virgin Islands. As I mentioned before, the yellow triangles represent mutton snapper spawning sites. The orange circles shown here represent different acoustic telemetry studies, some which were near shore, and others where fish were tagged at spawning aggregation sites on the shelf edge. We observed some very interesting movement patterns that provide critical information for management. For example, on St. Croix, the green arrows show the migration pathway of one tagged mutton snapper that traveled over 50 kilometers from the spawning site and was detected at receivers on Lang Bank and around Buck Island National Monument. This seems to suggest that there may be only one mutton snapper spawning site on St. Croix. More interesting, from several tagging studies around St. Thomas and St. John, we found that mutton snapper traveled over 50 kilometers. Mutton snapper traveled from home sites in bays, migrated to both spawning sites on the shelf edge, and moved between spawning sites during the same week. This indicated a lower level of spawning site fidelity than previously thought. Tagged fish seemed to visit both spawning sites, which were separated by about 22 kilometers. This means managers cannot just focus on a single spawning site for protection if the fish are using multiple sites. Let's take a closer look at the St. Croix mutton snapper seasonal closed area, which is closed for 4 months from March through June. The red triangles show the locations of our acoustic receivers along the shelf edge. At night, you can see up here on the upper panel, fish are located within the mutton snapper seasonal closed area. During the day, in the morning hours, the fish migrate and begin to leave the closed area swimming west. In the early afternoon, most fish are concentrated outside the protected area to the west. Since spawning occurs in the late afternoons, we suspect that this is the actual spawning site, which is outside of the protected area. It's interesting to note that the boundaries of the closed area were based on local ecological knowledge of fishers who use this area as their traditional fishing grounds during the snapper spawning season. Fishing occurs at night while mutton snapper are resting in their staging areas. Unfortunately, there is chronic poaching at this site during the spawning season, and we observe from 1 to 5 boats inside the closed area every night during the close season while we are tagging fish for our study. More work is needed to make this protected area more successful for mutton snapper. Before I summarize data on mutton snapper, I wanted to provide an example of grouper spawning site that has benefited from a small marine protected area in the U.S. Virgin Islands. One example of successful application of a seasonal closed area is the Germanic Bank, which I mentioned earlier for dog and cubera snapper. It was established in 2005 after a small aggregation of only 30 NASA grouper were discovered. The NASA grouper spawning aggregation that used to be in the area was completely fished out in the early 1980s, and the species became very rare and was listed as critically endangered. After the Germanic Bank was established, it took over 10 years to start to show any signs of population increase. Starting in 2020, about 15 years after protection, the NASA grouper population started to increase rapidly. This is mostly due to a large recruitment event that occurred around 2015. Last January, we counted over 1,000 NASA grouper at the Germanic Bank spawning aggregation site, and this has truly been a great success story. So now back to the mutton snapper story. I wanted to emphasize that mutton snapper are protected by multiple regulations in the Virgin Islands. There's a closed season and market closure from April 1st to June 30th for all of the USVI. This prohibits fishing, possessing, and selling mutton snapper, including recreational and commercial fishers and restaurants. There's also the mutton snapper seasonal closed area that I talked about on St. Croix, which is closed from March 1st to June 30th. However, despite these regulations, mutton snapper are still vulnerable during the spawning season. This is due primarily to chronic poaching inside the closed area. The actual spawning site and daily migration pathway is not protected by the area closure. And finally, if you recall, the month of July is outside the seasonal closed area, but mutton snappers still migrate there in large numbers during July. Some management considerations would be increasing enforcement to reduce poaching, adjust the boundaries of the closed area to include the primary migration path and spawning site to the west, and include the month of July in the seasonal closure. Due to the current level of poaching, there should be an effort to engage fishers in code management, including monitoring and research activities to increase their participation and compliance. I would like to thank everyone who came to listen to my talk, and I can take any questions now or later in the workshop. Thank you very much.
Thank you so much, Rick Nemeth. That was just a fantastic talk and huge amount of information in there for everyone to absorb. So our final talk, I'll introduce in a second, but I just want to say, give a big call to all of you attendees. Do please put your questions in the Q&A. We're going to have a 20-30 minute session after this. We really, want to have some interaction with you. And no question is too small. So, those of you who've never come across spawning aggregations and are just starting out, please ask us any questions. We're really open to discussion. So the final talk that you're going to hear is from Dr. Alec Hughes from Solomon Islands with some co-authors, and he's talking about managing around the moon, FSAs in Solomon Islands. Over to you, Alec.
Hi everyone, this is Alec Hughes from the Wildlife Conservation Society program in Solomon Islands, here to share a bit about our work on fish spawning irrigations in the western end of the country. Okay, very quickly, Solomon Islands is an archipelago located in the southwest Pacific. It's about a 3-hour flight northeast of Australia. It's got one of the highest population growth rates in the Pacific, with 80% of the population living in coastal areas. So there's quite a bit of a high dependence on fisheries for sustaining food and income, which is very typical of Pacific countries. The geographic spread of the country presents a challenge for top-down management approaches, especially for the Solomons, where enforcement capability is quite limited. So this highlights the importance of having community-based resource management approaches for ensuring better management of our resources. Recognizing the importance of these community-based resource management type approaches, the WCS program, which was formally established in the country in 2018, started working closely with communities in the Roviana and Marovo regions of Western Province in 2019 to support these communities with developing community fishery management plans for their locally managed marine areas. During our engagements and interactions with the communities, it became clear that there was quite a bit of concern from community members regarding fishing pressure on fish spawning aggregations, particularly from nighttime spearfishing. So with funding from the NOR institution in 2020, we were able to carry out some in-water monitoring of existing or established fish spawning aggregation sites for which historical data exists, and also to conduct local fisher surveys to try and establish where new FSA sites might be and to see how these fish spawning aggregation sites could potentially be incorporated into ongoing efforts to establish locally managed marine areas. Outlined in red on the screen here, we have is where the Marovo Lagoon is located in the Western Province, and this is where work took place that we'll be sharing. So very quickly, the Marovo Lagoon happens to be the largest saltwater lagoon in the world. As you can probably just make out from the map, the barrier reef system has several cuts or channels, passages, and promontories which help make it ideal for FSA site location. Early work by researchers has shown that traditional ecological knowledge is deeply embedded in the cultural practices, community structures, and the daily lives of Marovo people, particularly with those in coastal communities where the local fishers are well aware of where and when to harvest certain fish species. So using the fisher interview approach, we conducted several surveys around the Marovo Lagoon, focusing on several communities, and we will be sharing the outcomes of some of this work with the Perva community, which is located on Gataka Island, that red outline in the lower right of the screen. So zooming in further with Perva community, back in 2015 they first established the locally managed marine area with a no-take closure around Kicha Island on the right. In 2022 they expanded this to include the islands of Bulo and Male Male, and this was partly driven by the fact that the local fisher knowledge surveys we conducted highlighted up to about approximately 18 fish spawning aggregation sites that occur within the LMMA, and these sites were used by up to about 17 species of reef fish, including snappers, emperors, groupers, trevallies, and barracudas. With a better sense and appreciation of what spawning aggregations were all about, the community then decided to take a lunar approach with managing the spawning aggregation sites. So they put in place this 10-day closure for each month of the year, based on the period leading up to new moon, for which we know that a lot of these fish species tend to aggregate for spawning purposes. One species of fish that we are pretty sure forms spawning aggregations within the Perva LMMA is the Chinaman fish. So it forms these aggregations between the August to October months of the year, which is very similar to its very closely related species, the sailfin snapper. But this species, the Chinaman fish, we're not aware of any aggregations having been reported elsewhere in the country, so it's quite cool to know that this exists within the Perva LMMA. We've collected some gonad samples from the aggregating individuals, and they're all indicating that this is indeed a fish that's aggregating for reproductive reasons. So, looking ahead, there's quite a bit of work that still needs to be done on fish spawning aggregations in Solomon Islands, but we do want to be working closely with the Ministry of Fisheries and Marine Resources to put in place more appropriate management approaches to ensure better protection for fish spawning aggregation species. So this approach with managing around the moon or having a lunar-based sale and harvest ban is something that we need to explore more. We also need to encourage the inclusion of fish spawning aggregation sites wherever possible into LMMA design, like what has happened in Perva and other communities that we've been working with. And there's a need to verify more fish spawning aggregation sites, both those reported in Perva and elsewhere with our other data sets. And of course, we'd love to be able to get more in-water observations of Chinaman fish and other species within Perva, and of course, collecting gonad samples to better understand the reproductive dynamics of this species, in which for this we'll most likely have to team up with a research lab. Thank you guys, sorry I've run over time, but I hope that was informative. Thank you.
Well, that was fantastic. Alec Hughes, thank you very much. Sadly, I don't think Alec was able to make it, so he's not with us on the panel. We're now that's the end of the presentation. So we've now got a Q&A, questions and answers, and a dialogue session. We've got some great questions in the Q&A, so feel free to add some more. I think we've heard some amazing talks on really how to get down to researching your spawning aggregations and really documenting all the specifics. It's obviously quite complex. There are a lot of different methods out there. And we've really heard a lot from some amazing speakers. And we've got some of them here. Will is certainly here. He's one of the leads of this whole workshop, Will Heyman. And we've got Kevin Rhodes is now on the panel to be. Oh, we've got Rick. I thought you weren't going to make it. Fantastic. You are here. So we've got Rick Nemeth, and we've also got two key fishers who have been very involved in FSAs in the I think Belize and Florida. So forgive me because I'm from the other side of the world, but Western Atlantic, and that's Don De Maria and Mitch Lay. So welcome panelists, and I'm going to open the floor now just to I'm just going to go through the questions. I think that's the easiest thing. So we've got a question from an anonymous attendee who asks, in estimating abundance of FSAs, why other methods like swept area and depletion techniques have not been mentioned? Or have they got a number of limitations? If so, what are they? And we'll put a quick text reply in there, but I think he'd like to say a bit more, and maybe Kevin or Rick might have additional comments. So over to you, Will.
Thanks so much, and thanks for the great question. But quick answer is we're doing our best in 1 hour to provide a few tools, a few examples, a summary, and there's no way we can do it all. And yet, that's why we're putting this together, because we recognize there's lots of people all over the world that have all kinds of good things and techniques to add. So again, in the chat suggesting, hey, send us some links, send us some other things. We can use this platform to help share that information. In terms of the specifics on your question, I'm going to pass that over to Kevin and or to Rick.
Kevin, how's your mic? Are you there? Can you put your video on?
Yes, I'm actually here. I did. Can you repeat the question again, Melita? I'm not sure exactly I understood what the person was asking.
They were asking about what other methods, like swept area and depletion techniques, which have not been mentioned, what other methods are there? Or do those other methods have a number of limitations? And Will gave us a quick synopsis saying there's heaps of methods out there and we haven't got a lot of time, so we haven't covered everything. But I'd like to say I don't know that we'd want to use depletion techniques.
Understood. Yeah, I think
on a fish spawning aggregate. I don't know if you add. Yeah.
Yeah, I would agree with that. Yeah, I think in terms of the swept area method, that would probably be something that's more suitable to things like snappers that are water column oriented species. And certainly, you can use a variety of methods. There's time swims, there's point counts, there's fixed transects, there's the variety of methods. And really, it just depends on the species that you're trying to monitor and what information that you're trying to get out of it. So there's really no one method, one size fits all method. So I would say to any of the participants looking to get into FSA research to choose the method that's really right for you and do your investigations around all the different methods, and then pick one that works for you.
Great, great. Thank you. Okay, well, let's move to, we've got a couple of other questions here. So there's one from Denise Frederick. What are the critical issues that can bring about sustainable management for migratory or transboundary species between two different areas, e.g. one country and the other? And I think possibly somebody from the Caribbean might be good to talk about that, because there are large migrations for transient aggregations over there. So I don't know whether any of you, I don't know, Will, would you be able to pick that up? And maybe Mitch can say something.
I'll take a quick stab and then happy to pass it over to Mitch or others. I think this was something that we recognize. A lot of the countries in which I work, in this region are quite close together. We've documented migrations of snappers and groupers that go right from Belize into Mexico and back. And there's been a tremendous collaboration between collaborating countries, both at the government level as well as the NGO and the scientist level. There's something called the Mesoamerican Reef in the Western Atlantic, and several countries there are all working together on a regional aggregation project called MARFISH. There's also, importantly, the Western Central Atlantic Spawning Aggregation Working Group that has sponsored and developed a regional management plan for Nassau grouper and mutton snapper. And you'll hear more about that in Winston's session from Yvonne Sadovi. So there's a couple examples of where we're making some progress. Over.
And on that, Will, picking up on that, on the sort of governance piece, within the regional seas, there are these UN organisations like ROPME in the Arabian Gulf, RCOFI in the Red Sea, PERSCA. So these are the sort of inter-country forums where these issues are discussed and agreed, and knowledge shared.
Yes, and if I may just add, in the Caribbean, we have, in addition to the Western Central Atlantic Commission, we also have the Caribbean Regional Fisheries Mechanism, and we have a counterpart in Latin America as well. And they do work on management issues and including FSAs. So we do have a fair bit of collaboration there between countries. Thanks.
Great to hear you there, Mitch. Your contributions yesterday were fantastic. So I might ask you to answer a couple of other questions as well. Possibly this next one actually would be a good one for you. I don't know if you've managed to see it yet, but this is a question from Claire Thallis. How do you balance the need to publish research, including maps, to meet journal publishing time limits and ensure the data and research is recognized by government and conservation management practitioners against the need to keep the locations of spawning aggregations concealed for their protection? I think it would be very nice to hear an answer on that from Mitch, if you'd like to. But also I think Rick has done so much mapping around sites, and how certainly you're also on the SURFA board, the Society for the Conservation of Reef Fish Aggregations, where there was a very strong push at one time not to be very careful how you publish locations. So I think it would be very cool to hear something from you, Rick, maybe, or Will. Please put your hands up if you want to answer, but I thought Mitch would be great. Kevin, sure. So who'd like to go first to answer that question about concealing the location of FSAs when you publish them?
So I'll
Rick,
take this up for, okay. Rick, you can go first.
it's Mitch.
Oh,
Oh,
Okay. Yeah. Good morning, everyone. Yeah, I think it's really important, and it's been a topic of discussion for a long time. I know the SURFA websites reduces the number of significant digits on the GPS coordinates, so it's not that easy to find. The published spawning aggregation sites, so that's one method. But there are problems if you even the information from the research that's shared with local management, that can get out. And fishermen are very savvy, and if an area is not closed and they'd like to try to improve their catches before a site gets protected, oftentimes there's increased fishing pressure on these spawning sites, thinking that it's their last chance to actually catch fish there. So it's a very sensitive issue, and I think we need to work very closely with management to try to increase the time it takes to implement new management, or at least engage with the fishers so they understand the process that's moving forward, and to try to protect the species that are there.
Excellent point, and Rick, and really appreciate everything that you and SURFA have done on this. I guess one of the things that we've found is, we talk about fishers' knowledge and the importance of fishers' knowledge, but one of the things that we've found is not only are we kind of interviewing fishermen, but those fishers that are knowledgeable about these sites are often real important community leaders. So in our research, we've adopted them and brought them in as paid consultants and captains on the vessels, and they are really our cooperative researchers, and as well as government, and bringing people in. So as part of the process of the science, we've built this set of collaborators, leaders in the community, and they have been absolutely critical in moving the conservation process forward. Indeed, in Belize, we met with 8 or 9 of the key fishermen up and down the country with the minister, who said, "Okay, I believe the science, but I'm a politician. Let me hear from the real people." And so we brought all these fishermen together to meet with the minister right before he signed this historic legislation. So anyway, let me pass it back to you, Melita.
okay. I saw there was a bit of a hand up from Kevin at one point. Did you want to say something on that question, or should we move to the next?
No, I'll just make a quick comment. I think that, for the most part, we all learn and find or rediscover aggregations through fishers and through their knowledge. It's very, very, very rare. I can't myself think of a case where an aggregation was discovered without a fisher. And so these sites are usually widely known anyway. And so that's why it's so important to bring fishers on board in the research and share information with them, and to try to get them behind you. Many of them already understand that declines are ongoing and they want to participate in the management process. And so it's really not always so much about getting the fishermen on board. It's a lot of times getting the politicians on board, like Will mentioned. And so the political will usually comes from those stakeholders. And so, really the fishers are so very important, not only in the research, but in the management outcomes around the findings and around protections of these sites. So I'm just going to make that point there. Thank you.
That's great, Kevin, and excellent point as well. I think that's true. I think those of us who have worked on FSAs have all been shown them or taken to them by fishers. That's for sure. I think there was a lot of concern about ramping up commercial exploitation of them. And that was the one of the things about being a little bit careful about advertising where they are. And also, I will say that when in regions of the world, such as the Western Indian Ocean, where we have coastal areas are really dominated by artisanal fishing, and in some countries the pressure is now really high. So I don't think there's a lot of scope for increasing pressure on some of these species that form spawning aggregations. So we are fairly wary about that. And certainly when Jan Robinson and Jude Bijou and I were doing that work, it's over 10 years ago now, we were pretty careful about sort of advertising them. Anyway, so moving on, I'm just going to turn to, actually, it's the final question that's in the Q&A. Can one do, it's from an anonymous person, can one do research to find spawning periods and aggregation areas by just looking at gonads of the fish sold at the market and asking questions to the fisher of where they were caught? And I'm going to have a first stab at that because the talk I gave in this session was looking at those sorts of techniques, but then I definitely want to hand it over, and I think probably Kevin will be great to add to that as well. I think the great thing about looking at gonads in the market is one of the things I stressed was what's telling you the fish is spawning is that the ovaries are hydrated. Well, there's lots of names for that. Running right, the eggs are extruding out of the fish, and you can't miss it. And that hydration period is very short, so it's guaranteed. If the fish have got hydrated ovaries, then they are spawning. It's right in their spawning period, but it's not telling you it's from an aggregation. And obviously it's not telling you where they were either. So if you ask the fisher where were they caught, then you can perhaps find the aggregation site, if there is an aggregation site. But that none of that has verified that it is a spawning aggregation site, because that's where you need the other criteria as well, that you certainly heard a lot about that from Will's talk and also from Kevin. So I think I'll hand over Kevin maybe next, if you'd like to add something to that.
Thanks, Melita. I think that pretty much covers it, really. The eggs, the landing sites, the markets will give you the first indication that the spawning aggregation is formed somewhere, and then it really is then gaining the trust of the fishermen over time to try to lead you to that site. Some of the sites can be quite far away, quite offshore. It's not easy just to follow a fisherman to a site and have it discovered. You have to work with the fishermen, but the markets, the landing sites, and sometimes just, number of boats fishing at a single site will give you all the indications you need to start your research. And research is not an overnight thing, and finding these locations and getting to the management and enforcement part can take quite a long time. So you have to be patient in your pursuits and realize that it's not going to be an easy or overnight thing to get to that end result if that's your goal.
Yeah, exactly. No, thanks for that, Kevin. I think key point. I don't know, Will, if we've got another question I just want to relay to the whole audience, but Will, did you want to add anything in there?
No, but I do see Rick has his hand up.
I think I was going to do the question he's answered. Yeah. Do you want to talk about it, Rick? So Khalid Mohammed put a question in the Q&A asking about fish aggregation devices. How can we benefit from FADs in spawning aggregation or fishing spawning aggregations? I think he's asking. Rick has answered that in text, so you should all be able to see it. FADs, fish aggregation devices, typically used for pelagic species. So in this workshop, we're really talking about demersal species. Yeah. So we weren't really sure whether you're asking, can FADs be used to help aggregations recover or to increase catch rates? And I know that this workshop doesn't enable, because there's so many people, attendees, to answer verbally, but maybe Rick, you can add something else there, but we weren't totally sure. And if you feel unsatisfied, write us another question. There's a few minutes left. Maybe that's the way to go. Did you want to say something, Rick?
Yeah, I was going to actually, on the previous discussion about location of spawning sites. Like I provided in my example, sometimes the fish use these staging areas. And so the actual spawning site is quite a bit a distance from where the fish are resting, either right after spawning or before spawning. So we found in our research that catch rates actually go down during spawning because the fish are interested in reproduction and not in feeding. And so when they leave the spawning site is when you actually start getting higher catch rates. And so during the validation process, it's important to make sure that you combine the fisher knowledge with other types of assessments to actually locate the sites if any kind of spatial management is going to be put in place. Yeah, that's all I was going to say. Yeah, thanks.
Actually, whilst you're there, can I ask you a question? Because I've loved your work with all those different areas, the mapping and how fish use aggregation sites. It's just incredible work. But I did wonder, how applicable is it to a variety of taxa, particularly the taxa we're interested in here? And was it largely done with groupers? I guess I didn't quite pick that up. So particularly that that external ring around the sites that you had was sort of hundreds to thousands of kilometers. And I thought, you know, I've worked on species that don't move that kind of distance. So but form transient FSAs. So I was curious to know, was that very applicable to where you work in the Caribbean and the islands, etc.? Just to put you on the spot.
Yeah, so I think the farthest area, the catchment area, is really if you track all the fish that are at a spawning site to their home sites, they both snappers and groupers go tens to hundreds of kilometers away. And so if you're on an island shelf, that could include the entire island shelf, say in the Cayman Islands or St. Croix. We're on the St. Thomas is located on the Puerto Rican shelf, so it's much larger. And we estimated swim speeds and distance time that the fish was away. And you can kind of estimate how far that their home sites might be, and they could be closer or they could be further away. So it really varies, and the work that I've done with Kevin as well depends on. Like in the Pacific, you have these atolls where the fish move into the reef passages to go into the lagoon. And so the way they travel is much different than if you have this large flat shelf area where they could go north, south, east, or west, or whichever way the shelf edge is. They can go very many different directions to disperse from the spawning site. So that really kind of directs how the management area might be placed. Groupers we found, even though they seem very sedentary, they were moving 5-10 kilometers every day. They were leaving the spawning site, returning the next morning, getting closer and closer to the aggregation, and then spawning started at around sunset. And then around midnight, they dispersed 5 to 10 kilometers and then came back again. So that cycle, I think, partly helps young naive fish find their way back. And these groupers are vocalizing that entire time that they're moving away and to and from the site. And I think that increases participation in the spawning sites by the groups of fish that are around there. So I think there are some similarities in many of the species. And that's one of my goals. I'm kind of working my way through different species to understand those differences and similarities. And definitely the snappers and their related species move a lot more and much more active during the day, moving up and down, many kilometers, even just before spawning. So it's a very interesting science, and it's not easy, but I think we're getting a lot of information from this that can be used as general guides. And I think one really important thing, if you're protecting a fish that might be critically endangered, the protection around that spawning site at multi-species sites protects many other species. So I think that's a great benefit of spatial closures.
Yeah. Well, I think that's a great, fantastic reply. Thank you for that. And it's a great segue into management, which we'll definitely touching on that in the next session. But I think Ken Lindemann has a good question for Kevin on customary tenure in protection of FSAs. I can pass over to you, Ken, and then we should wrap up because the next session's got to start soon.
So this will be fast, but it could, of course, take hours, Kevin. In the Atlantic, many of us are fascinated by the concept of customary tenure from Johannes and others. Do you have a favorite example from the Pacific of customary tenure being deployed over time to protect FSAs? Thank you.
Yeah, well, customary marine tenure basically is where ownership of the reef by a village or a chief or a clan takes place, and they make decisions, management decisions around those closures. And so that's very common to a couple of states of Federated States of Micronesia. It's throughout Melanesia, Solomon Islands, Papua New Guinea. Alex earlier talked about the Piava community as locally managed marine areas, but those are all okayed by the chief. The chief has the final word on whether those closures are going to take place. In terms of a favorite place, I don't have necessarily a favorite place, but I do know of several, and I do know that they've been effective. You know, where we have the luxury in the Pacific of having ownership of the reef, that's a very, very important management tool. Chiefs will often open and close an area as well based on how the fish populations are doing. So they're not always a permanent closure. And so that's something important to recognize about customary marine tenure as well. It's adaptive, it's flexible. And so it can be a very effective way to manage your fishery. Thanks, Ken.
Great question and great answer. And I think that topic's going to come up again in the next session. So we're going to close this session now, session 3, and have, I think, a very short, like 1 or 2 minute break, bathroom break, before we move into session 4. Thanks so much to the panelists and the and all the speakers and the attendees. See you very soon.